Well, welcome to the knowledge room, the concepts of life sciences and biotechnology. Today we are going to understand something about the protein and its structure and how this is relevant to its function. Protein catalyzes the metabolic reactions, it powers cellular motion, forms macromolecules, rods and cables, those maintain the structural integrity of hair, bones, tendons and the teeth. In nature, forms follows function. Structural variety of the proteins reflects the sophistication and diversity of their biological roles. Newly synthesized polypeptide mature to perform the biological functions and it becomes a functional protein that folded into specific three-dimensional arrangement that is also called a conformation. There are post-translational modifications that add chemical groups or remove transiently needed polypeptide segments. Genetic or nutritional deficiency impedes protein maturation, results in deleterious effect to the health. For example, Creutzfeldt-Jakob disease, scurvy, Alzheimer's disease, bovine spongiform encephalopathy, which is also called a mad cow disease, scurvy, a nutritional deficiency that impairs the protein maturation. Configuration versus conformation. It seems that these both are same, but by definition, configuration is the geometric relationship between a given set of atoms that is part of the protein. Those distinguish L from the D amino acids. An interconversion of configurational alternative requires breaking covalent bonds, whereas the conformation refers to the special relationship, means in the three-dimensional space, the relationship between the atoms in a molecule. Globular proteins are compact, roughly spherical, and ovoid in shape. And the axial ratio, that is the ratio of the shortest to the longest dimension of not over 3. Whereas the most enzymes are globular proteins whose large internal volume provides ample space in which to construct cavities of the specific shape, charge, and hydrophobicity or the hydrophilicity required to bind substrates and promote catalysis. By contrast, many structural proteins adopt highly extended conformation. These fibrous proteins possess axial ratios of 10 or more. The lipoproteins and the glycoproteins contain covalently bound lipids and carbohydrates. Examples, myoglobin, hemoglobin, cytochromes, and many other proteins contains tightly associated metal ions which are very important for their functions are termed as metalloproteins. Protein perform complex physical and catalytic functions by positioning specific chemical groups in a precise three-dimensional arrangement. So the conformation is important. The polypeptide scaffold containing these groups must adopt a conformation that is both functionally efficient and physically strong. If we go by normal permutation and combination, a typical polypeptide can adopt 10 to the power 50 or more distinct conformation. Folding into the conformation appropriate to their biological function would appear to be even more difficult. The modular nature of the protein synthesis and folding are embodied in the concept order of the protein structure. These are the four in nature. The first one is the primary structure, that is the sequence of amino acids in a polypeptide chain. The secondary structure, folding sort of 3 to 30 residues, contiguous polypeptide segments in a geometrically ordered unit. The tertiary structure, the three-dimensional assembly of a secondary structural unit to form larger functional units such as the matured polypeptide and its competent domains. and the quaternary structure, that number and type of polypeptide units of oligomeric proteins and their spatial arrangement. This is the example of the four structure. The first one is the primary structure, which is just the sequence of amino acids. The second is one of the secondary structure, that alpha helix. There are the beta pleated sheets and there are the beta tones and there are the omega loops. So out of the four types of the secondary structure, here it is only the alpha helix is depicted because this is part of this protein, the globin of the hemoglobin. And this is the tertiary structure, that is made up of the one protein, the beta chain of the hemoglobin, and this is the quaternary structure that is associated to the heme iron. Thank you.